Hello everyone, welcome to today's topic, which is one of the most complicated topic and have made it as simple as possible. So today's session is on autophagy. So in this session you will learn the definition of autophagy, what are the various types of autophagy, causes and its role in health and disease. What is autophagy? Autophagy is a process in which cell eats its own contents. So in Greek, auto means self and phagy means eating. So a cell is eating itself. How is it being done? Cytoplasmic materials or cargo. So cargo means nothing but the cytoplasmic materials which are getting delivered to the lysosomes for degradation. So what is the most important organelle which is involved in autophagy? It is lysosome. So it is one of the most important bit. So organelle involved in autophagy is lysosome. And autophagy is also a type of programmed cell death. So you remember what is the other programmed cell death? It is apoptosis and various new forms of cell death like necroptosis and ferroptosis. What are the genes involved in autophagy? So these are known as autophagy genes or ATG genes. What are the various types of autophagy? We have macroautophagy, microautophagy and chaperone mediated autophagy. So in macroautophagy what happens is there is delivery of cytoplasmic cargo to the autophagosome which then fuses with the lysosome for degradation. So as you can see here, in the initial stages there is membrane formation that is known as phagophore formation. Later on it is fused to form a autophagosome. So this autophagosome later on fuses with a lysosome to form autophagolysosome. So the classical feature of macroautophagy is autophagosome formation. So next coming to microautophagy. What is microautophagy? In this microautophagy, there is no autophagosome formation, but the cytoplasmic cargo is directly engulfed by the lysosome through invagination. So as you can see here, no autophagosome is formed in the microautophagy and there is lysosome. So in the lysosome, small invagination is formed as shown here, so which engulfs the cytoplasmic material. So that is microautophagy. No autophagosome formation, just by invagination of the lysosome, the cytoplasmic material is getting engulfed by the lysosome. Next is chaperone mediated autophagy. In this what happens is, these chaperones, which are heat shock proteins, specialized proteins, these recognize and translocate damaged proteins across the lysosomal membrane. So as you can see here, so these are the damaged proteins. So these damaged proteins are recognized by the chaperone proteins, which are heat shock proteins, and they are getting delivered to the lysosome, and there in the lysosome they are translocated and the damaged proteins are lysed by the lysosomes. So it is also autophagosome independent. So only in macroautophagy autophagosomes are formed. No autophagosomes are formed in microautophagy and chaperone mediated autophagy. But the common thing for all the three types of autophagy is lysosome formation, that is lysosome degradation. Ultimate organelle which is involved in autophagy in all the three types is lysosome. Coming to the process of macroautophagy. So in macroautophagy, already I told you there is a membrane formation, that is a phagophore is formed. The green colored thing which is shown here is a cytoplasmic cargo. So around the cytoplasmic material which has to be degraded, around that a membrane is formed known as phagophore. Okay, that also known as isolation membrane, and this isolation membrane is mainly formed by the endoplasmic reticulum. Also it can be formed by the mitochondria or the plasma membrane. So once isolation membrane is formed, next a double membrane vesicle is formed around the cytoplasmic cargo. So here as you are seeing here, it is a double membrane vesicle. So this is autophagosome. Now this autophagosome fuses with the lysosome to form autophagolysosome. So what is happening with this? The lysosomal hydrolytic enzymes are getting released into the autophagosome, resulting in degradation. That is, autophagolysosome is formed where because of the hydrolytic enzymes the cytoplasmic material is degraded. So this is the process of macroautophagy. So already I told you, in microautophagy and chaperone mediated autophagy there is no autophagosome formation. It exclusively occurs only in Macroautophagy. Coming to the process of macroautophagy, how does it occur? It occurs in 6 steps. It is by, first step is initiation, followed by nucleation, then elongation, maturation, fusion and degradation. So why this autophagy or macroautophagy is occurring? Because of nutrient deprivation and depletion of growth factors. When there is nutrient deprivation and depletion of growth factors, what happens is AMP kinase is activated. Once AMP kinase is activated, it acts as sensor and it activates a group of proteins known as ULK1 complex or initiation complex group of proteins. What are they? It consists of ULK, ATG13, FIP200, ATG101. As I told you, ATG is nothing but autophagy genes. Okay. So once these complex is activated, what it does, it helps in the formation of pre-autophagosomal structure. So this ULK1 complex further activates another complex known as Becklin-1 complex by phosphorylation. What is the function of this Becklin-1 complex is, it helps in the nucleation step of autophagy. It is constituted by Becklin-1 mainly and ATG14L, VPS15, VPS34. So Becklin-1 complex has a role in nucleation, whereas ULK1 complex has a role in initiation. So ULK1 complex, once it gets activated in the initiation step, later on it activates Becklin-1 complex by phosphorylation. So in the nucleation step, what is happening? So there is phagophore or isolation membrane formation. So already I told you there is isolation membrane formation by the endoplasmic reticulum. Also, it is done by the mitochondria and the plasma membrane. So, here a phagophore is formed all around the cytoplasmic cargo. After the nucleation step, next step is elongation step. So, in the elongation step, there are specialized proteins known as 2 ubiquitin-like conjugation systems. What it does is, it helps in the covalent linkage of phosphatidylinositol to microtubule associated protein, that is LC3. So there is covalent linkage of PE to LC3 resulting in PE-LC3 complex as shown here. So what is the speciality of this PE-LC3 complex is, it forms a membrane around the target material, around the cytoplasmic material. Membrane is expanded. Already it is formed in the nucleation, but in this step it is expanded further. So as shown here, okay, the membrane is getting expanded, and the speciality of this PE-LC3 complex is, it is a useful marker for identifying autophagy. So marker of autophagy is PE-LC3 complex. So whenever this complex is increased, it indicates that autophagy process is going on. And the next step is maturation step. So this maturation step is also mediated by this PE-LC3 complex. What is maturation is, in the elongation step, the membrane is elongated, but in the maturation step, there is closure of the membrane to form a closed double membrane vesicle known as autophagosome. So this is autophagosome. Okay. So next step is fusion step. In fusion step, what happens? There is fusion of autophagosome with the lysosome. Okay, so to form autophagolysosome, and the final step is degradation step. In the degradation step, what happens? The lysosomal enzymes, it is rich in hydrolytic enzymes, so it digests the cytoplasmic contents and molecules, and what happens after that? The molecules which are broken down, these components are recycled for utilization by the cell. So this is degradation. So this is a mechanism of autophagy, how it is going to be done. So so this is mechanism of autophagy, but autophagy also has to be, should be a checkpoint for autophagy. How it is done? By the inhibitors of autophagy. What are the inhibitors of autophagy? First inhibitor is mTOR. Okay, it is mammalian target of rapamycin which inhibits autophagy. How is it inhibiting autophagy? Is it is inhibiting the ULK1 complex, or also known as initiation complex. So mTOR inhibits ULK1 complex and thereby inhibits autophagy. Another inhibitor of autophagy is Becklin-1 complex is inhibited by BCL2, which is a pro-apoptotic protein. So BCL2 inhibits Becklin-1 complex. Another drug is there known as rapamycin. Okay, so this rapamycin specialty is it inhibits mTOR. Okay, so it has therapeutic indication in various neurodegenerative disorders and cancers. Why? Because rapamycin initiates autophagy. How? mTOR is inhibiting autophagy, whereas rapamycin is inhibiting mTOR. So ultimately with rapamycin what happens? mTOR gets inhibited and that's why autophagy gets activated. So this is the gist of the mechanism of autophagy. First is initiation step, which is mediated by ULK1 complex, and ULK1 complex is activated by the AMP kinase, and ULK1 is inhibited by mTOR, whereas mTOR is inhibited by rapamycin. Next is nucleation step. In the nucleation step, there is formation of phago-4 or isolation membrane by the PI3K complex or Becklin-1 complex. Third step is elongation. There is elongation of the isolation membrane, which is done by the PELC3 complex. And fourth step is maturation. In this, a well-formed vesicle is formed around the cytoplasmic cargo. It is known as autophagosome. And fifth step is fusion, where this autophagosome fuses with the lysosome to form autophagolysosome. Last step is degradation. Because of the hydrolytic enzymes, the cytoplasmic cargo is degraded. What are the various types of macroautophagy? Macroautophagy can be selective or non-selective in nature. In the selective autophagy, depending upon the material which is getting engulfed by the autophagosome, it is given the name. First one is mitophagy. Means, as you can see in the diagram, what is inside the autophagosome? Mitochondria. So that is mitophagy. In the mitophagy, the target organelle is mitochondria, and the genes involved are Parkin gene and PINK1 gene. This mitophagy is very important in many neurodegenerative diseases. So, as you go into the systemic pathology, you will learn various neurodegenerative disorders which are associated with accumulation of abnormal misfolded proteins. So, all those mechanisms are because of macroautophagy and that too mitophagy. So, next thing is spexophagy. As the name indicates, spexo means peroxisomes. So, the material present inside the autophagosome is peroxisomes. Then is reticulophagy. So, the material present inside is endoplasmic reticulum. Then agriphagy means it is protein aggregates. Xenophagy means intracellular microbes are getting engulfed by the autophagosome, that is known as xenophagy. And last is lipophagy. Lipid droplets are getting engulfed by the autophagosome. So, this is selective autophagy. Coming to microautophagy. So, up to now we have dealt with macroautophagy. Coming to microautophagy. So, what is microautophagy? Already I gave introduction for the microautophagy. It is cytoplasmic material are directly enclosed by the lysosomal or vacuolar membrane without the formation of a double membrane vesicle. So, no autophagosome formation. The cytoplasmic material is directly engulfed by the lysosomal membrane by just invagination of the membrane. So, as you can see here, the cytoplasmic material is directly presented to the lysosome. And how it is entering into the lysosome? By the invagination of the lysosomal membrane. So, here you can see, because of invagination, the cytoplasmic material enters into the lysosome. No autophagosome formation. And the last type of autophagy is chaperone mediated autophagy. So, in this chaperone mediated autophagy, it is specialized form of autophagy where it identifies the misfolded proteins. So, this misfolded proteins are identified by specialized group of proteins known as chaperones, that is heat shock protein 70. So, after this misfolded proteins are getting bounded by the heat shock protein 70, what happens is they are recognized by the lysosomal membrane protein complex known as LAMP2A. So, it is a membrane protein present on the plasma membrane. So, LAMP2A is present on the plasma membrane. Once this misfolded proteins bind to the heat shock protein 70, this LAMP2A gives a way for the entry of the misfolded proteins into the lysosome. Okay. So, once these misfolded proteins are entering into the lysosome, then what happens? There is unfolding and translocation of the misfolded protein which occurs inside the lysosomal lumen, and later these misfolded proteins are degraded. What are the various causes of autophagy? Nutrient and energy deprivation. In nutrient and energy deprivation, nutrient shortage inhibits mTOR. So mTOR is inhibitor of autophagy, and nutrient shortage activates ULK1 complex, thus initiating autophagy. So when there is deprivation of energy, what should happen? Autophagy has to be initiated. That is, there should be autophagy in our body. So in that process, ULK1 complex is activated by AMP kinase. Whereas when there is nutrient or energy excess, autophagy has to be inhibited. So in that process, what happens? mTOR is activated, which further inhibits autophagy. And the second cause is accumulation of damaged or misfolded proteins. In neurodegenerative diseases like Alzheimer's, Parkinson's, Huntington's disease, misfolded proteins are getting aggregated, and later on they are undergoing autophagy by chaperone mediated autophagy. When there is reduced insulin or insulin like growth factor 1 signaling, there is decreased mTOR activity. So when there is decreased mTOR activity, autophagy is getting increased. Rapamycin, while telling the mechanism, I told you rapamycin is direct mTOR inhibitor, that is mammalian target of rapamycin. So it pharmacologically induces autophagy, and this mechanism is being used therapeutically to treat some cancers and intracellular pathogens, that is xenophagy. So by xenophagy, some intracellular pathogens are getting eradicated or removed by autophagy. This autophagy occurs as part of normal embryogenesis. It removes unnecessary organelles during tissue remodeling. What is the role of autophagy in health? So it helps in the recycling of components. Why should we have recycling of components? Because it functions as a survival mechanism to maintain integrity of cells during stress. Okay, so when we do fasting, there will not be any energy to our body to undergo metabolism. So how is that energy derived? If no food is given, because of recycling of the components. Okay, so it is a survival mechanism. And how is atrophy occurring? It is because of this autophagy only. When there is severe nutrient deprivation, what happens? There is atrophy of the cells, and that is done by autophagy. And the second role of autophagy in health is it helps in the recycling of the proteins, and it destroys the damaged organelles. Okay, so once there is damage to the organelles, they have to be removed from the body. So that process is done by the autophagy. So the turnover of organelles like the endoplasmic reticulum, mitochondria and lysosomes is done by the autophagy. And protein aggregates. So there are certain protein aggregates getting formed. So what is getting done with autophagy is this protein aggregates clearance is being done by the autophagy, which results in which is responsible for various stress, various disease states like neurodegenerative diseases. So by autophagy, when there is clearance of the protein aggregates, it can prevent certain neurodegenerative diseases. And elimination of pathogens. So intracellular pathogens are getting eliminated by the autophagy. What is the role of autophagy in human diseases? So it has a role in cancer. Autophagy can both promote cancer growth and act as a defense against cancers. Okay, so it can be pro-carcinogenic as well as anti-carcinogenic. And it has a role in some neurodegenerative diseases. Defective autophagy, it leads to accumulation of misfolded or aggregated proteins. So the examples of neurodegenerative diseases like Parkinson's disease, where alpha-synuclein aggregates or Lewy bodies are formed. Huntington's disease, where mutant huntingtin accumulation is getting done. Alzheimer's disease, where beta amyloid and tau protein aggregates are formed. And certain infectious diseases like mycobacteria, Shigella, HSV are degraded by autophagy. Macrophage specific deletion of autophagy 5. What happens with this is, when there is specific deletion of autophagy 5, it increases the susceptibility for tuberculosis. And it has a role in some inflammatory bowel diseases like Crohn's and ulcerative colitis. When there are single nucleotide polymorphisms in the autophagy related genes like ATG16L1, it leads to Crohn's disease and ulcerative colitis. So these are the questions which are framed on today's topic. I request all of you to solve these questions and give your valuable answers in the comment section. I hope this video has been useful to you. Thank you, and meet you in the next session.